Welcome. This is part two about bacteria. So, bacteria part two. Last time we learned about a couple things about bacteria, and I want to review those with you from way back. Remember that they're prokaryotic, unicellular, and they can have multiple energy sources, right? Which we talked about, heterotrophic or autotrophic. And of those, they can be photoheterotrophic. Some can use light energy and eat other things, which is kind of neat. Some don't even need light. Those chemiautotrophs use those chemicals from deep in the ocean. The nucleoid is the region in the bacteria cell where the DNA is. And remember, there are different types of oxygen requirements for bacteria. Some are completely anaerobic, which means they don't need oxygen. Oxygen will kill them. Some are completely aerobic, and those facultative ones can use both oxygen or oxygenless environments, depending upon what is around. Now, this one, part two, we're going to talk about bacteria reproduction and ways in which their DNA can change, the genetic changes within bacteria. You read about this on Monday, hopefully, but bacteria reproduce through a process called binary fission. Fission, this word here, you might have heard of like nuclear fission. There's also nuclear fusion. Fission is kind of a breaking apart, whereas fusion, if you hear that term, things are being joined together. So binary fission, we're splitting apart. Here's a nice little picture of bacteria fizzing in action. You can see the two that are being separated here. But really, it's nothing fancy. It's just making a copy, an exact copy of itself, or a clone. Here's a nice little picture. You're going to start, the DNA replication just like in any kind of cell or cell cycle. And after it replicate, you're going to go right to cytokinesis, and it's going to divide. There is no mitosis here because there is no nucleus, remember. So just a random fact there. But again, that's binary fission, just the way they reproduce exact copies of themselves. Nothing fancy there. But what is interesting, to me at least, is the genetic changes, how the DNA within a bacteria cell can change. And there's three primary ways: transduction, transformation, and conjugation. But before we get on to that, there's one little piece, a little extra nugget for you. Another term is a plasmid. Lots of what's going to happen here you're going to end up forming plasmids within these bacteria cells. And a plasmid is extra genomic DNA. Right? The DNA that's come from outside. It's not part of its normal genome, normal set of DNA, and that nucleoid is separate. So here you have your normal bacteria DNA. It's in the red, and the plasmid, here multiple plasmids, are in blue. And it's these plasmids that are a lot of what we'll be talking about next, and how the DNA in the bacteria cell changes. You're going to have a lot of plasmids. Antibiotic resistance is often due to a plasmid. This bacteria cell picked up a plasmid and now is no longer destroyed by the antibiotic. All right, let's go through these three ways in which DNA, the genetics of a bacteria cell, can be changed. First one, transduction. All right, transduction is when we have a virus cell injecting bacterial DNA into a bacteria cell. Remember from the viruses, learning about viruses, when a virus infects a cell, it attaches, like this guy here, and all it does is insert its genetic material into the cell. But what will happen is a cell will be infected by a virus. Here, like number 2 is infected with a virus. And what will happen as the viruses are assembling themselves into viral particles before they lyse the cell and explode it out, they manage to pick up bacteria DNA inside the creation of this new virus instead of the viral DNA or RNA. And then when the virus is released, it goes to infect another bacteria cell, and instead of injecting the viral DNA and making copies of viruses, it actually injects just the bacterial DNA, and that bacterial DNA then can incorporate itself into the genome of the bacteria inserted, or become a plasmid. So that's transduction. The virus doing the work for you. And so again, transduction, the virus mistakenly inserting bacterial DNA into bacteria, so that viral DNA. All right, next up, transformation. So bacteria also have the ability to pick up DNA, foreign DNA, or just any kind of DNA, prokaryotic DNA, from the surroundings. And what we'll have in this picture here, you have bacterial cell die, maybe even through that virus that destroyed the cell, you get chopped up bits of DNA. But there's just DNA floating around in a sample, and the bacteria cell can allow that through its membrane. And once again, that bacteria cell can incorporate it into its own nucleoid or form plasmid. A transformation, and we'll have another presentation on genetic engineering and stuff later. But scientists use this ability of bacteria cells to pick up foreign DNA to transform and manipulate bacteria into making things that scientists want them to make. We mentioned in the last bacteria, part 1, that bacteria now can make insulin because of the way scientists have changed them, and this is through transformation. They find the target gene for insulin, and they make it, and then you put it into, and we'll get into more of the process later, but essentially they're inserting a gene that they want into the bacteria cell, and the bacteria cell then does what the DNA tells it to do, and in that case it's make insulin. It's kind of neat. And then the final way to change DNA is through bacterial conjugation. And this is important, and I remember correctly, I think it's somewhat newer in terms of their understanding of bacteria and what they can do. This is pretty cool from the bacteria's perspective, but it's really this transferring DNA from one bacteria cell to the other without real loss of the DNA in the donor cell. So it's just copying DNA and sending it over to another bacteria cell. This is the best I could find. Thought I could find a joke, but it's probably not that funny. Anyway, you can chuckle. Hey, have some DNA. This is a micrograph of what's going on. You have this bridge that's connected, and what will happen is one bacteria cell will form a bridge extending to the other bacteria cell. I accidentally clicked the mouse there. And it's going to send DNA over through this bridge. And here's kind of more of an animation. I think this is the DNA that's going to get sent over. So you have your DNA that's being sent over, and it can be the plasmid or it can be the actual nucleoid. It depends on the event. But you have this bridge attachment, and it's always one direction. So the donor cell is going to take the DNA and send it along through the bridge to the acceptor cell, and it's going to form a plasmid within that cell. This is how antibiotic resistance can be spread from bacteria cells. If you have that plasmid that's antibiotic resistant, and you run into another cell, and you're able to transfer it to that new cell, and you end up with more bacteria cells that are antibiotic resistant. Or you can also pass on the virulency of it. If you have a gene on here for a toxin, you can pass it over, and now an innocuous bacteria cell then becomes harmful. But again, that's bacterial conjugation, just sending DNA over from one bacteria cell to another. And the amount of DNA that's sent depends on this bridge. See, it's kind of thin. It can be kind of weak, and it's going to send it over until the bridge is broken. Most of the time, it's not going to get the entire genome, and most of the time, not even the entire plasmid, but some's going to be passed over. And this is how bacteria then can change or evolve through time. So again, those three ways of change are transduction, the viral mistakes, through transformation, picking it up from their surroundings, and bacterial conjugation, which is the direct transfer of DNA from one bacteria cell to another without loss in DNA, with no loss in DNA from the donor cell. All right, don't forget, subscribe and like my video. Enjoy your day.